Do some quick intros, and then I'd like to tee up this topic a little bit so that as we listen to the presentation by Jeff and Mark, we'll have a sense of what we should really be looking for in this presentation. First, let me make some apologies for Ruben, Dr. Carbonel. He literally was in transit to this meeting up until yesterday and wound up with a family emergency that required him to change his plans and not be here. So he sends his apologies. If you're wondering why we have an empty chair on the podium here, it's not the Air Force routine. Let's do a little bit of intro, and then we'll get back to the topic at hand. For myself, I'm Andy Skibo. I'm EVP of Operations at MedImmune, and now as we fully merge our operations into AstraZeneca, Regional Vice President for Biologics for AstraZeneca, which is the same role, plus I'm responsible for engineering and real estate worldwide. I've had the pleasure of being in pharma since my first project was 1976 for Eli Lilly in Ireland. About 80% of my work has been offshore. I joined Genentech in '82, and I've been with the bio side of the industry for what must now be going on 40 years. Been in operations, engineering, validation, have at least 6 product launches under my belt. That's the piece of the market that we always aspire to. Building plants is fun, but launching a product is the only thing I think really that gets us up in the morning and puts us to bed at night. If I talk a little bit about Jeff Odum, Jeff is the Director of Operations for IPS based in their Morrisville, North Carolina office. Jeff has been in the business for over 20 years, probably worked on $2 billion worth of plants for any number of the major manufacturers. That's one of the huge advantages of having some of consulting industry under your belt. About 40-50% of my career was in that side, is you get to work on a lot of different projects for a lot of different owners, and it gives you a constantly renewing perspective of what the industry did and what the industry now requires. And then last, if I talk about Mark Witcher, Mark also has a great deal of experience in this industry. He's on the technical consulting side, process side, bio side for IPS. Prior to that, he worked in manufacturing at Covance, and prior to that, he worked in manufacturing at Amgen. We were chatting briefly at lunch, and we gave up adding up project values when we hit north of $10 billion. So you have a great deal of project expertise on this stage. But more importantly, it isn't the history of what was done 10 years ago or 15 years ago. It's a sense of, and the three of us volunteered to present this session because it's near and dear to our heart, is this industry is in a dramatic change, a flux. This is one of the inflection points in the industry for any number of reasons. I'll give you a couple to think about. Then when Jeff and Mark do a tag team on the presentation, you can focus in on where they are. Why do I say this is an inflection point? I lived through a number of changes in this industry. Back in the 85, 86 time frame, if you could get 0.3 of a gram out of a 15,000-liter bioreactor, you considered yourself as just working miracles. Many of the big battleship plants, the 4 by 15,000 liter, the 6 by 15,000 liter, traditional, if you think of bulk bio, that's the type of plant you think about. MedImmune owns one. Amgen, Genentech, Biogen, IDEC all own at least 1 or 2, sometimes 3 and 4. They were based in the late 1990s, early 2000s on titers of 1.5 grams per liter, with 3 being considered outstanding. Today, titers push 7 and 10 grams per liter. Even an average contract manufacturer, where they develop the process for you, would probably target 2.5 grams per liter, but a good process is 7 to 10 grams per liter. Since these plants take 6 years to design, license, to concept, design, construct, license, and bring online, For many companies that invested, the typical plant 4 or 5 years ago would have cost $550 million. Today that plant will cost $800 million. For many of those plants designed to do 1.5 to 3 grams per liter with titers now at 7 to 10 grams per liter, you have an automatic stainless overcapacity of, is it 3x, is it 4x, in some cases 7x. There are pipelines out there that literally could require a capacity of 2,500 liters as the top bioreactor instead of 15,000. But that's a change, because if you ask the industry 4 years ago, would another one of these battleships ever been built, probably all 3 of us 4 years ago would have said it would be the rare board of directors that would approve that. 3 years ago, a number of us began adding up what was happening in biosimilars, what was happening in the fact that biosimilars are not going to displace a lot of legacy products because, at least in the United States, clinical trials have to be run to prove equivalency. If you have to spend $800 million getting a bioequivalent proved, why would you not just go and spend $1.2 billion and get your own novel approved if you have a novel pipeline? So you don't yet see in the United States a march to biosimilars. If you look at pipelines across all companies, these pipelines are beginning to mature, and it doesn't matter which company you talk to, whether it's MedImmune, over 100 assets in play, near-term readouts of north of a dozen products in the next 2 or 3 years. That's unheard of in this industry. If you ping Genentech, if you ping Amgen, every single one is in a position of suddenly having to reevaluate, do they have enough bulk capacity? And that's driven by a number of things. One, those legacy products are not going away. So if you're going to continue to produce X hundreds of kilos of a legacy protein and its yield is 1.5 grams, you need a battleship. There are new products in the pipeline that have a combination of lower yields, they're just difficult to produce, and they have a high protein demand. Almost every company I talk to has at least one of those. If you have a product that's going to have a very high protein demand and you just can't get its yield above 1.5 grams, you need a battleship plant. And there are a few biosimilars that will make it across the finish line. They will, by definition, require a battleship plant because they're typically based on the best parallel to the original model, the best molecule, the best parallel to the original molecule is the same process or similar to the old process, so low titer. So park the thought. Battleships are here to stay, and that's a part of the mix. But the other part of the mix is, what about those products that require only 4 runs at 7 to 10 grams per liter? A change in the industry going forward. 10 years ago, 7 years ago, you would run one product in one of these large plants, and you might change over products every 18 months. So the fact that changeover time might require 3 weeks out of a month was immaterial because you only did it once every 18 months. There are some plants that ran the same product for 5 years straight because the plant was built for a particular product and the company didn't have 5 products. I'll give you some real-life examples, but MedImmune is not unique. I'll just quote examples so that this isn't just theory. If I look at a plant the size of Frederick, 4 by 15,000 liter bioreactors, and I go back 5 years, we'd make one product every 18 months and switch over and perhaps run 2 products on a turnaround once every 18 months, and that's all that we would do. So turnover time was immaterial, maximum yield was the driver. In 2020, thanks also to a partnership with Merck where we share capacity, and I'll give a few minutes on that and then let Jeff and Mark do their thing. By 2020, that site will run at least 10 products. They will range in titer from less than a gram per liter to 10 grams per liter. They will range in lot size from 40 to 50, 15,000 liter bioreactor lots to 4 at 2,500 liter bioreactor size. We will probably have 6 to 7 turnovers a year. So suddenly an 18-day turnover time is a killer. If you do 6 of those, you've lost half the capacity of the plant just due to turnover time. So what does that mean? In 2020, the plant of the future, and we're blessed with the fact that we happen to have a small 2 by 2,500 liter bioreactor plant for one of our smaller first products. The site will have a small plant at 2,500 liters, fixed stainless. It will have at least the existing 4 by 15,000 liter bioreactor. It may have 6 by 15,000 liter bioreactor. Rather than one downstream purification line for each of those packets of fermenters, we will probably have a combination of 5 because downstream is now the limiting capacity. In addition to the fixed stainless, we will definitely be looking at single-use technology because one of the ways of minimizing turnover time, especially if you only have to run 3 or 4 lots of a product, is to not do it in fixed stainless. What that requires us to do, and what I'd like to ask you to do as your homework item, even though it's late in the day, is as you listen to Mark and Jeff and they discuss the possibilities of what can be done, think of what you will have to be doing as you decide, what do I build? What do I seek out? What would I have to have online in 4 years, 5 years, 6 years? Because that's the length of time that you're making these decisions. And the answers aren't monolithic. They're not simple anymore. They're not single. Go build a fixed stainless plant of this capacity. Go build quick turnaround single-use technology. Go build a battleship. Go build 2,500 liter. The answer is not simple. It's going to be a mix of all of the above. And then the last thing that makes the decision more complicated is the industry has finally broken through this. It was more emotional. At one time it was regulatory affairs. It used to be very hard to get a bulk plant cross-licensed for someone else's product, either yours or using a CMO for your own product. You could do that with fill finish, but in the late 80s, early 90s, the concept of deep tank mammalian cell was so novel that it was really hard to get a CMO or contract manufacturing operation approved. That's no longer true. Also, there was great pride of needing to control capacity. That need is still there, but given the financial state of the pharma industry, no board of directors is going to approve a $200 million, a $500 million, a $600 million investment until it's too late to actually go build the investment. You won't get such an approval until at least phase 2B and ideally phase 3. So capacity sharing is now a fact of the industry. Medi's presented on this many times. We did this with Merck at Frederick. We will make a lot of Merck mammalian cell culture products because we have an ocean of such stainless available. They will make our microbial products, and every pipeline includes some microbial candidates, even in this day and age, because it happens to be the best technology for a narrower slice of your pipeline, so it's not going away. We don't want to build one of those plants for 10% of our pipeline, so with this two-way street with a partner, we can share. So add all that together, and then as you listen to Mark and Jeff, listen to what a plan of the future can be, and at the end of this, we're going to get into a question and answer session and try and explore some of these issues. But the encouragement is, don't look for any one train of thought as the right answer, because almost definitely there is no single right answer to the question. I'll turn it over to Jeff.
Thanks, Andy. One of the challenges about this time slot is we're the only thing standing between you and adult beverages. So keep that in mind as we go through the slides. As Andy said, as we talk about for the next few minutes what can be, keep in mind that there is no right answer for everybody that fits every scenario. My guess is that a lot of you who are sitting in the audience work with organizations, or you have clients that will fall somewhere in between the concept of the traditional stainless steel battleship that Andy talks about, and then some of the concepts that we're going to address over the next few minutes. But the focus is really one that things are changing. As we look at where we are today in the biotech industry, there are a lot of challenges, or to some they're seen as great opportunities. We are seeing that there are an increase, significant increase in potential products in the pipeline. We're also seeing that a lot of the anticipated markets in terms of patient population and what will be seen as viable to support the global needs are much smaller. And as the science continues to advance, we believe that that trend will continue. One of the things that has been the Achilles heel of a lot of facilities over the past decade has been the concept of utilization. You put a tremendous amount of economic capital into a facility, and when you sit down and actually look at the statistics from a utilization standpoint, you find that you are not very efficient. And so one of the challenges that many companies, many of our clients in particular, are looking at is how to improve the utilization in terms of not only multi-product type scenarios, but also the idea of multi-phase manufacturing, and we'll talk about that in a little bit more detail, but the idea that why can't we, from the standpoint of a manufacturing philosophy and a regulatory philosophy as well, be able to manufacture from the clinical stage all the way through commercial manufacturing in essence the same asset, and be able to do that over a long period of time for a wide range of products. We want to look at throughput. For a lot of us that are sitting in the room, when we talked about the concept of fast in terms of what the expectations were a decade ago, today the speed is 10x, because as Andy mentioned, the number of products, your ability to turn a facility around on a dime is becoming more and more of not only a value but a necessity. It really becomes a fundamental part of the business model. And so we're seeing that more and more companies are having to look at that particular aspect of how a facility is going to operate. And it has a tremendous influence on how you put a facility concept together in the very beginning. And change is here. We are going to continue to see a dynamic change in the industry because of the science, because of the new types of products, increases in titers and yields, and that change is something that we are going to have to be able to address much better than we have in the past 2 decades. So if we look at what has gotten us here, well, a lot of the advances have come not only from the science but also in the advances from an engineering standpoint. As we mentioned, the productivity of the process itself has dynamically increased and will continue to do so. The advent of single-use equipment for the first time really since its inception, introduction into the industry a number of years ago. From vial to vial, for many applications, you can find a single-use solution for your unit operational needs. Now, there are still some challenges that are there with regards to scale and certain types of product lines, but we have that ability, and that is something that we didn't have in the past. This also gives us a tremendous advantage from an infrastructure standpoint. We're looking at a fundamental shift in what a facility will actually look like. We sort of joke about it, but if you are in the business or you're one of these people who loves to take advantage of the opportunity to go on plant tours for any given local event you may have at a professional society, they're going to become sort of boring because what you're going to see in a lot of instances in the future is not a lot of nice, shiny stainless steel with a lot of glass and something that really speaks of high tech, but you're going to see containers that are going to hold bags, and there's going to be tubing, and they're just not going to be real exciting from the standpoint of the tour. But it will definitely define what becomes efficient and allows companies to really increase utilization. And around all this is the concept of segregation, and we're going to talk about that in a little more detail in just a little bit. But as you begin to define your approach from a manufacturing perspective and how you look at segregation is really going to play a key role in what facilities are going to look like. I'll also mention that from a regulatory perspective, we are starting to see a shift with regards to what the regulatory bodies, in particular the FDA, is beginning to look at. They realize that this paradigm shift is occurring and that the ways and means of inspections and the way things were looked at in the past are going to have to change. And so they, too, are a part of this transition process that we're seeing in the industry today. So as we talk about this concept of facilities, and the idea is to maximize the opportunities that a company has, whether you're a CMO, whether you are manufacturing a mainline biologic, or in some instances it may be part of some program that, for example, is run by or administered by federal agencies where pandemic response is the key. The ability to adapt and have flexibility in your manufacturing operations is critical. The one thing that in this model that's important is to minimize those lost opportunities and to make sure that as you look at all of the inputs and define unit operation parameters, that not only is there an understanding of what the product attributes have to be, but in terms of the process itself and how you look at those parameters that are key and essential to meeting those needs, that we look at flexibility, we focus on the efficiency and throughput of the facility, and the idea is to, like I mentioned, minimize those lost opportunities and therefore minimize potential losses as well. So the challenges then sort of fall into some very general categories. The ability to manufacture in multi-phase is one area, again, probably not for all, but many companies are looking at this as an opportunity to minimize capital expenditures, to do away with the fact that I may have to have 2 different facilities or possibly 3 to get me from the clinic into commercial phase manufacturing. As Andy mentioned, the advent of biosimilars across the globe is going to present new challenges and issues for a lot of organizations as they begin to focus on how best to bring those products to the market. Changes with regards to the different therapeutic families of products. We're seeing, again, with the advances in the science, more opportunities for different product types. The ADM, or Advanced Development Manufacturing model, is one right now. It is mainly the focus of different agencies within the federal government. We are involved in some projects of this nature, but it puts a new light on a different business model, and that business model is very, very focused on speed and flexibility. When a pandemic event occurs, one of the things that you are wanting to occur very rapidly is the ability to identify the threat and respond in a way that protects the patient population as large and as fast as you possibly can. So fast is good, but faster is now the expectation. We are seeing this both in terms of expectations from a manufacturing standpoint. We're also seeing it in terms of the way that clinical trials are being designed and executed. And we also have found that if you look at this ability or this desire to minimize tech transfer from site to site or location to location, that too is playing into this, that more and more facilities will be able to handle not only development work, but also commercial manufacturing at the same time. So these enablers, we've mentioned some of these already. We're seeing a great increase in yields. More and more, the idea of plug-and-play, skid-based type unit operations is becoming more common practice. The advent of continuous processing continues to grow. We see that in a lot of instances, the minimization of downstream unit operations is becoming more and more of a focus, not only from a flexibility standpoint, but from a risk perspective as well. And the efficiency at which those operations are being ran is greatly improving. Single-use systems are becoming more commonplace. We're seeing that not only are more and more companies beginning to focus on how they can implement these systems, but we're also seeing more of a focus from a regulatory standpoint on better understanding and analyzing risk. When you talk about single-use systems and you talk about the ability to validate system closure and looking at risk to the product in single-use systems. So we're seeing this shift occur across the board. And a lot of this is really being driven by regulatory changes that have occurred over the past few years as we've seen the advent of Q8 and Q9, the new process validation guidance that was issued in 2011. Have sort of set the stage from a regulatory standpoint on how companies are beginning to look at conceptualizing their designs and developing manufacturing solutions for what they're going to do over the next decade. I'll let Mark talk a little bit more in detail about the segregation strategies and some of the models themselves, and then we're going to open it up and begin to deal with questions from the audience.
Thank you, Jeff. In the facilities design, one of the primary issues is the separation of the different processes and different unit operations that are occurring within the facility. So there is a separation imperative that must be met. To realize that imperative, you need to implement different segregation strategies, of which there are a couple. You can use facility time, and therefore you have a single unit operation or a single product within a facility for any given period of time. But more recently, single-use technologies has developed a capability of having closed process systems. And another very important segregation strategy is the procedural controls which can be used to manipulate the two physical segregation capabilities that I discussed. In terms of primary physical segregation, you can have single-use facilities in time. In other words, you have basically campaigns going through where you separate the unit operations and the different products within space and time. These are run very independently. However, they consume a great deal of resources within the facility. Recently, the closed systems have become popular, and there is a considerable amount of effort going into validating the use of single-use systems as being a widely used method of separating the different processes. However, there is more or less a real issue that has to be addressed. It doesn't appear to be common understanding in the industry. We really need a process validation paradigm when you talk about validation of closure. There is the design to be closed, and this is what the single-use industry has been working on for a considerable length of time, and I believe they are there. There is the qualification that you have single-use closure, and this is tested and qualified to close. But when the FDA states that you must validate closure, they're really talking about the third hurdle, and that's the stage 3 or verification. And that is answering the question, was the single-use systems closed at the time they were implemented? That places a tremendous burden on the systems that you're used to establish that the single-use systems remained closed. And there is a tremendous burden of proof which is developing, and this is what the industry is beginning to wrestle with. The current trend in the industry is to work with large operating spaces, and this provides they take care of the opportunities for manufacturing for multi-product by commingling products within large open spaces. If we take processes, they can quite often be divided into what can be called logical operating units, and this is how they are divided up. And the number of logical operating units for a process can be between 2 to 7 or 8, depending on the process needs and what kind of segregations are required. If you look at multi-product, then you would have more than one process or product within the space at the same time. This brings up a number of issues. While you can get reasonable utilization at this, you begin to identify some problems with resilience. Single-use systems are fairly complicated. A single-use bioreactor is a complex tubing set with up to 25 and maybe even more different tubing sets coming off of the tube. There's a lot of ways to mess that up. So you must deal with a system in which you have single-use problems. Throughput is impacted by this type of design because there's a considerable number of interactions between the different products within an operating space. And that problem can be exacerbated if you look at multi-phase manufacturing. In other words, one of these processes might be early phase clinical while the other process might be a commercial manufacturing. You also have reliability issues in terms of the failure interactions that you have to deal with. The likely solution to that is to begin to look at facilities in which you have a matrix of rooms, none of which are dedicated to any specific operation, but allows you the flexibility to move these skid-mounted processes in and out as the availability of the facility is realized. You have an opportunity for both multi-product and multi-phase because all of the logical operating units are all separated by individual space within the facility. The resilience and reliability is enhanced because any problem that you have with any of the unit operations is localized. It can be dealt with independently. But one of the features that I think is the most attractive is that you have an enhancement of throughput and utilization because each one of these processes can be operated independently. In other words, you may roll in the inoculum step well before you would roll in any of the downstream processes, and so you would be able to segregate them. It would also give you even additional flexibility if there are hold steps between the logical operating units within the process. And this also provides greater operability in the sense that you have an incoming corridors shown in the light green. That allows you to operate the facility in a unidirectional mode, but it also allows you to operate it in a bidirectional mode depending on how your good manufacturing processes work for the specific processes. You have an outflow of material, so when that you destage one of the unit operations, you can take it out the out corridor. It also gives the ability, in the case of a problem with one of the processes, to implement a unidirectional flow and thus preserve the integrity of the incoming corridors. So this type of facility provides a considerable amount of flexibility and is one that is particularly applicable to the advanced development and manufacturing, or ADM models, where you would want to bring the processes in very quickly at scale in order to begin clinical manufacturing and shakedown runs, and then go straight to commercial manufacturing to manufacture the millions of doses that are required to supply in case that particular product is released by the FDA under an EUA mechanism. So here we have a facility that has a considerable amount of flexibility. It has high utilization. It has high throughput because you can move things in and out independently of each other and other processes. and it also provides you a great deal of efficiency in terms of its operation. So I believe for some manufacturing applications, this type of matrix type of facility is likely to be one that will be explored in the future. And with that, I will turn it back over to Andy.
Great, thanks. Thanks, Mark, I appreciate it. And we're going to, since Dr. Carbonel is not here today, I'm going to do double duty and be a part of the panel as well as being a bit of a moderator here. So forgive us, we'll try a tag team flexible experiment. Let's open with, you've heard a lot of different concepts here, and concepts that are very new, concepts that are traditional. Let's start with any questions from the audience. We had a number submitted earlier, and I can go to those, but I'd be first interested directly from the audience.
Hi. I had a question about whether you see a difference in manufacturing facility of the future for mAbs versus vaccines. How do you see those two being different, and what would they be like?
Very good question. And